investigation is on strategic environmental assessment process for sustainable environmental plant biology and biotechnology towards sustainable biotechnological development. Sustainable biotechnological development is a kind of development that meets the needs of the present generation without compromising the ability and efficiency of the future generation to meet their biotechnological requirements. So this has basically three pillars: environmental stewardship, social inclusion and economical growth. The objective of the study is pertaining to the investigation to establish strategic environmental assessment process. In many of the projects, biotechnological projects, plans, programs, policies and legislative actions, the environment and sustainability are considered in planning and decision making process that could dominate biotechnological, economical and environmental factors in the world's plant biology and biotechnological projects. So the main focus I have taken is the insect resistant BT cotton and the BT cotton varieties, prediction assessment of environmental health effects. Environmental health impact assessment is a systematic identification and evaluation of the potential environmental effects, human effects of the proposed biotechnological seed cotton projects. For example, insect resistant bollworm, the seed cotton, genetically modified organisms, as well as naturally pigmented color cottons. So these have necessary for the conduction of the environmental health impact assessment process, that is a systematic identification and evaluation of the potential environmental and human effects of the proposed projects, plans, programs with respect to the physical environment, chemical environment, biological environment, biochemical environment and social economical environment of relative, total and human environment and environment. So you can just see that this is the seed cotton. So before 2001, the seed cotton was based on only the using the fertilizers and pesticides, the grown seed cotton, and the insects were insecticides were used, pesticides were used in the field. But after 2000, genetically modified seed cotton, that is using the BT cotton, biotechnological cotton, have been market in most of the countries in the world. My investigation is more than 100 varieties I have taken. So sustainable plant breeding technologies have been identified and evaluated with systematic study. Cotton seed cottons can be made resistant to drastic environmental stress and infections. Plant breeders across the globe have developed various short staple, medium staple, long staple and extra long staple hybrid cottons. Insect resistant BT cotton, naturally pigmented color cotton, new varieties that is called Bacillus Thuringiensis Jenny seed cotton that are called Kapas, or cotton plants that is genetically modified to contain the endospore, so that produce toxicants of the Bacterium BT and to be resistant to certain insect pests. So I would like to highlight this toxicological effects have been taken place in the field. Not only in the field, it carry forward in the textile engineering field, spinning field. So the toxic characteristics is a hazardous substance. The toxic is a hazardous substance. That endospore toxins Bacillus, that is called, we call it as a BT load. So nearly 90% BT load is at the seed cotton, because it produces toxicological effects, harmful effects, and can be defined as that because of its quantity and characterization and concentration, physical, chemical, infection characteristics that may cause significantly contribute to an increase in mortality or cause an increase in serious irreversible or incapacitating reversible illness or pose a substantial present or potential hazard to human health and the environment when they are improperly mitigated by means of the environmental health impact assessment process for the BT cotton. BT load is nearly 80 to 90%, should be reduced to 1%, because 1% is the threshold limit. 0.1 ppm is the maximum toxicological permissible limits in the field, but it goes mere 100 to 150 ppm, which is more than the permissible limits, 100,000 times more than the permissible limit. So it is necessary to mitigate the environmental problems by means of these Bt cottons presently grown in the world. Before 2001 there was not the big issues, but now this is necessary for us to consider the strategic environmental assessment process. And this is how the investigation have been taken place. That is called product and process approach model. And Bt crops are transgenic crops that are genetically engineered from the DNA, deoxyribonucleic acid, which is a substance carrying genetic information of Bacterium, Bacillus, Thuringiensis. Bt crops are transgenic crops that produce the same toxin as the Bacterium, Bacillus, and that is the seed cotton plant cells produce crystal insecticidal proteins in the plant cell, thereby protecting the crops from pests. And the environmental impact of transgenic Bt seed cotton in context to plant biology and agricultural science with respect to environmental sustainable air, water, land due to Heliothis process that is investigated. Therefore, case study and check aspects of Bt seed cotton are discussed here. The Bt seed cotton plants contain one or more foreign genes are obtained from the soil dwelling bacterium called Bacillus and are transgenic seed cotton plants. Bt seed cotton has grown exponentially, accounting for 90% of the seed cotton growing in our country. Since its introduction in 2002, Bt seed cotton has benefits to the environment from a considerable percentage of reduction of chemical pesticides, insecticides, but a considerable percentage of loss of land, air and water, ecological biodiversities as per the law of land, air, water, interaction, interrelation, ecological theory. So India have investigated when I was Cotton Technological Institute in Matunga, Bombay. So Bikaner Nirma hybrid cotton was investigated. Seed cotton grown in the field at present in our country is Bt seed cotton. That is a biotechnology modified, genetically modified organisms, which is different totally. It's more than 80%. That is wild ancestors is called a genetically modified organism or genetically modified pest and insect unwanting vegetation resistant seed cotton Kapas variety and produces a pesticide and insecticide combating bollworm. And so during the production, the production function is given. Y is equal to function of x1, x2, x3 to xn, where x1, x2, x3, x4 these are the breeding inputs. Y is the breeding output. So by means of these Bt toxins genetically modified that have been investigated 200 varieties and inputs such as pest, insect resistance, Bt toxic proteins and herbicides, each harmful to different insects, pests, and the toxic proteins are produced by the crop plants or worm ingested and become dead worms that are Bt cotton and or bollworm resist seed cottons that have the potential of 90% Bt load, toxic load on the environment. So this is the kind of attention for the present investigation towards the sustainable development process, which is a kind of development that meets the needs of the present generation without compromising the ability and efficiency of the future generation to meet their own needs. And the results also have been extended for the environmental health pollution aspects that are there by using the chrome composite leather clad rollers which are used in cotton ginning industries, and it has the environmental problem of chrome absorption in the lint cotton. So biogenetic manipulation of Bt seed cotton. So two pollutants have synergistic effects. The Bt seed cotton is genetically engineered with Bacillus, a biotoxins that comes from soil bacterium. The Bt that was isolated from soil is available to seed cotton as an organic pesticide. The Bt genes produce a protein that cuts into the guts of insects and pests and renders the seed cotton resistant bollworm insects. The biotechnology for the control of bollworms that is made available in the seed itself. The resultant seed cotton variety having ability and efficiency to produce Bt protein and defend insects and pests that also have considerable environmental poisoning toxicological effects of LD, lethal dose 50% test organisms, it is nearly 100 to 150 ppm, which is beyond the permissible limit. So deoxygenation and reaeration, how in the field the deoxygenation and reaeration, deoxyribonucleic and ribonucleic simultaneously taken place. So the bottom point, it is a very anaerobic condition. So where the dissolved oxygen is very much minimum, so which have the potential toxicological effects. So the investigation is pertaining to the psychological health effects on the people in the vicinity. So the EHAA, the project proposal for the genetically modified BT seed cotton to assess the toxicological effects by visual inspection as per environmental psychological health readings on workers and nearby residents. Nanotoxicology resulted as 1000 millionth standard order to neurosis and psychiatry towards sustainable nanotoxicological development. So the strategic environmental assessment process has been investigated for making much earlier decisions in planning and decision making process than environmental impact assessment process. So environmental impact assessment process, which is a need of the BT seed cotton and the genetically modified organic biotechnological aspects. So which is environmental impact assessment is must in this particular plan, project, policies, even the legislative actions, which is a systematic identification and evaluation of the potential environmental health effects of the proposed projects, plans, programs, legislative actions with respect to the physical environment, chemical environment, biochemical environment, then social economical environment, cultural environment of the total human and environmental environment. So the investigation to identify the study of the environmental problems existing with the present chrome rollers which are employed in cotton roller ginning industries and design the new rubberized eco-friendly roller gins. So this is a process how the McCarthy ginning is taken place, folding the fiber and remove the seed from the seed cotton. So materials mammals, earlier walrus, the blubber was used, and in USDA, United States Department of Agriculture, Gillam, who has done extensive work on the properties of gin roller covering materials for cotton roller gins. So this is the cotton roller gin. The gin roller is an important element of the cotton roller ginning process to remove the fiber from the seed. From the seed cotton, seed separated, lint cotton separated. Lint cotton contains the toxicological load, that is the excretion of the bacteria of the BT load, called the toxicological load is 90% in this lint cotton, and chromium also absorbed in the cotton roller ginning process in our country, especially in our cotton ginning industries in our country, double roller ginning machines. The chromium absorbed is large effects. So this is a double roller gin, how the ginning is taken place, and the lint cotton, we call it is the lint cotton, and this is a seed coming from the machine, and this is the lint cotton is baled about 179 kilo, taken to the spinning process into the spinning industries, ginning industries, then oil spilling industries, then spinning industries, and the grid is the gin roller investigation device, which I have done it in during 2000. Before BT cotton was developed, I have investigated the chrome composite the materials. So this is the chrome composite leather clad rollers which are used in cotton ginning, Indian cotton ginning industries, which has the environmental problems. So chromium pollution is nearly around 13,000 to 14,000 ppm mg per kg that have contaminated the lint cotton beyond the permissible limit. So nearly around 2000 ppm this contains, the lint cotton contains 2000 ppm. So it means it is beyond the eco-friendly level. So the level is very high. So that's why we need to pay attention on the environmental health impact assessment. So the lint cotton get contaminated with the chromium pollution, the two chromium 3 and chromium 6, which produces cancer effects among the people who use the lint cotton and the fabrics and fibers that include the occupational and non-occupational exposures in the ginning industries, biotechnology, and it also one of the incidents for the COVID-19 also. And this sort of how the chrome composite leather clad rollers which are used in the cotton ginning industries, and how the maintenance practice taken place. So this is 180 mm diameter, and after one season 3 months this is reduced to 110 mm. So nearly 60 to 70 microns that get absorbed into the lint cotton and beyond the certain environmental friendly limits. So this is the engineering drawing of the CCLC roller. This is the seed grid showing, and the beater going on beating the seed cotton from the seed cotton remove the fiber from the seed, and the roller wearing investigation have been taken place. The roller reduced to about 40 mm, nearly 50 micron per hour is contaminated into the lint cotton. So percentage of the material removed from the roller is 43.8%. And the roller wearing data have been investigated. Chromium toxicity results, that is affecting the various organs in our body, skin, nose, lungs, kidney, liver poisoning, and brain disorders, then lung effects that lead even the COVID-19, the biotechnological disease. Biotechnology is a cotton disease, chronic lung disabled disease to the workers, and acute and chronic toxicity, and the chromium pollution level is monitored here. The single roller ginning machine, and how the rollers are due to the dust producing grinding action in the process. So this powder, how the powder will have the effects in the process, and the various roller washers, and how the cotton workers exposed with this lint cotton and cotton dust biocenosis in the industries. And this is a synergistic poison because of the BT load. Because of the biotechnological genetically modified organic lint cotton have got nearly around 90 to 100 ppm of the toxic loads. So this also is called a synergistic poisoning. It is augmenting effect. I is the augmenting effect took place. So just like the photochemical smoke in Los Angeles take place, the same manner it is a photochemical action. So lung effects take place in an occupational worker in an Indian industry, and this is the roller engineering industries. The workers, the women workers, getting exposed with the BT, and the atomic absorption spectrophotometer analysis to find out the chromium pollution levels and BT levels. So we consider that BT is the organic method, but it also produces the toxicological effects, environmental toxicological impacts. So that is why I have proposed in this project the strategic environmental assessment process, and that too after COVID-19 world, post COVID-19 world, it is necessary to conduct certain projects, especially nuclear power projects, especially the BT genetically modified biotechnology, biology projects. This need environmental health impact assessment process, which is also systematic identification and evaluation of the potential environmental health effects. So that's what we have seen, the deoxygenation reaeration study, deoxygenation Cedar-Phillips oxygen sac curve, which is shown in the earlier presentation, and that need the environmental health impact assessment with respect to the physical environment, chemical environment, biological environment, biochemical environment, cultural environment, and social, cultural, social economical environment of total human environment. So human environment impact is equal to PAT, that is the population multiplied by affluence multiplied by the technology. So if it is a sustainable technology, then it will lead to the sustainable development. So if it is not unsustainable technologies, previous presenter mentioned about the unsustainable word. So it also lead as like production function y is equal to function of x1, x2 to x3 to xn, where x1, x2 are the input element, y is equal to output element, output divided by input is equal to efficiency. So this way the quality focus the degree of excellence with which the seed cotton to be the investigated how to be considered. So with this I would like to conclude my presentation. Some of my reference I would like to show. These are all some of the references I have taken. Taken. Then gastric, lung and oral cancer prevention through hazardous environmental impact assessment process, which was published in Lab Lambert by myself. I am the author of the book. And the other one is concerned the properties of covering materials of rollers used in cotton roller ginning industries, unsafe chromium contamination and pollution from cotton ginning industries, and development of eco-friendly alternative. This is also published by Lab Lambert. And Bisnosis and coronavirus disease impact assessment process, and strategic environmental assessment process, and environmental impact assessment for sustainable development. This book also published by Lab Lambert in Germany. And this is another important book, strategic environmental assessment process for sustainable life sciences, that is evaluating cotton gin walrus roller covering materials for cotton double roller jeans. Walrus was the first material used in 1940 by the Gillam and engineers, and the walrus was extinct after considerable years. So UK and USA have investigating the new rollers. So that is the scientific improvement of the properties of gin roller covering materials by Gillam, scientist. He was a very renowned scientist working in United States Department of Agriculture, USA. And some of the publications is sustainable agricultural seed cotton development case study and check, environmental health impact assessment process for the sustainable agricultural seed cotton crop research, environmental and sustainability health impact assessment process of BT Bacterium, Bacillus, Thuringiensis, seed cotton of genetically modified organism, GMO for sustainable development. That is a development that meets the needs of the present generation, that is 30 years period, without compromising the ability and efficiency of the future generation to meet their own needs. So with this I thank organizers Magnus to provide me an opportunity, and thank you very much.